And now let's try to wrap up and summarize everything and see that we are really all okay with all the things we covered. Let's begin with putting everything in context. So we saw many different tests and all kinds of measurements that we could make. So here we have a summary of everything according to the kind of the tests that we saw and what they're good for. So we saw that for two numerical variables, we could use correlation tests, so Pearson's or Spearman's. We could use regression. For numerical versus categorical, we saw that we can compare means, so we can compare it using a t-test or a u-test if we have two groups. Now, u-test, like we said, it's not, strictly speaking, not exactly comparing the means, but most of the time it's close enough, so I put it under this category, even though it's a bit different. We saw that we could use ANOVA if we have multiple groups. And if we want to compare the entire distributions, we could use Kolmogorov-Smirnov's. And then for categorical variables, we saw that we have the odds ratio and risk ratio, optionally with a confidence interval if we want to get a measurement of the strength of the association. And if we want a p-value, we can use a Fisher's exact, which works for the 2x2 case, or a chi-squared test that would work for any table under more stringent assumptions. And finally, we spoke about multiple testing correction. We saw that we could use Benferroni correction or two versions of the FDR. Now let's try to take all of that and see if we can actually apply it in context and see what kind of test we can use. So feel free to join, and yeah, let's see what you think about these cases. So let's say that we want to test whether nucleus and cytoplasm proteins have different degradation rates. What does it seem to you like? OK, yeah, we could use a t-test. Right, because we have two kinds of proteins. So we have two groups, and for each of them, we want to compare some numeric measurement, which is the degradation rate of the proteins. Now what about the second case? We want to test if sugar consumption is associated with cardiovascular diseases.
Yes or no, or sugar consumption as a...
Sugar consumption is something continuous.
OK, so here are all the different answers. And of course, it depends how we decide to model it. So like you said, we could model it to be something continuous or to be something categorical. So for example, what you suggest?
Correlation if we consider the spectrum numeric values.
For both values. So both sugar consumption and the disease you model? OK. So how would you model the disease as a continuous variable?
There's all sorts of scores for cardiovascular diseases, or I don't know, maybe it's less suitable.
OK, any more ideas? So we see we can use association test. Sorry?
Maybe a chi-squared.
A chi-squared. So under like what categories?
So like low, high, medium consumption of sugar, and sick or not sick, or different kinds of cardiovascular diseases.
OK, yeah. So we can build a 2x2 table and then have Fisher's exact, or a more nuanced table with more categories and then have a chi-square test. What other options? Fisher's, yeah. What other?
If t-test, if we take, population of cardiovascular disease and one not, and the sugar consumption can be continuous.
Exactly. So we can model the disease as two categories or more. And then we can look only at sugar consumption as the numeric variable. And then it's like comparing sugar consumption between groups, and we can use a t-test or u-test or something like that. So here you can see that actually we can use pretty much any test. It all depends on how exactly we model it. What about the third one? We want to see if exercise is associated with life expectancy.
Correlation.
Sorry?
Correlation is some sort of Fisher or Spearman or even a linear model.
OK, yeah. So again, we could measure exercise by some measurement, say a number of hours per week of exercise or something of this sort. And life expectancy is for sure something numeric. So yeah, we could do some correlation tests. What about the next one? We want to see if some Go annotation terms are enriched in the list of genes associated with ALS risk. I heard Fisher is exact.
Chi-square?
Chi-squared. Okay, so how would you define the contingency table?
What's
So
expected?
sick or not sick, and
Okay.
even I would say even for Fisher, not chi-squared even. So sick, not sick, and
You mean-
assume there's like
You mean-
non-enrichment, so something is distributed equally across, should be the same for those genes maybe?
So I want to make sure I understand. So you say sick or not sick. You mean people? So you start counting people that are sick or not sick?
Like people with, oh okay, so no.
It's
that
genes.
doesn't work.
It's a gene list.
What is the thing we want to perform, to analyze? What is the entity we want to analyze?
And the amount of genes or coterms in,
Go terms.
So we want to analyze genes. They are like the entities we want to say something about, not people. So what kind of, how would you design it?
Try permutations to do it.
How would you do permutations?
Get a list of genes and random, see what annotation terms you get with randomly assigning a list of that number of genes and see how many you get those specific terms and see if that's by chance or not.
So you want to count how many Go annotations you get by chance? Or those specific Go
I guess those
annotations?
specific ones.
Which specific ones? How would you determine which ones?
That's a good question.
Like the question is some Go annotation terms. So you need to check which one of the terms
Okay.
is enriched with this group.
Let's say that I want to get a list of Go annotations which seem to be enriched with ALS risk.
If you have a list of genes, okay, so the list is 1,000 genes.
Okay.
Random 1,000 genes, and see if you have an enrichment for some, certain annotation. Then, if we do it with annotation, we can see if the enrichment frequency of some annotation is significant or not.
Okay, but what would you change on each permutation? Like, okay, let's say for simplicity
You
say
try
that.
different genes, and you want to see to compare genes that are associated with ALS to a random list of genes.
Genes. Okay. So how would you get the list of Go annotations which are enriched?
I have the list of genes.
So let's say I have a Go annotation. I have a Go annotation which says, I don't know, DNA binding, okay?
Okay.
It's a
Now
Go
I have
annotation.
list of genes that are associated with ALS.
Okay.
Count how many genes have Go annotation of these 1,000 genes have this this Go annotation of by DNA.
Mm-hmm.
Right? Now we take random
Okay, got it.
genes and see how many.
The same one.
Okay, so what Ray says is we can. So let's say that we have 1,000 genes which are associated with ALS, and we have, I don't know, 200 genes which have some Go annotation, say DNA binding. And now to see if that's like, and then we have some overlap between them. I don't know, we have, let's say, 30 genes which have both annotation. We want to see whether 30 is a lot. We can randomly perform this this assignment many iterations over. So assigning 1,000 genes with ALS annotation and assigning 200 genes with this Go annotation and see whether 30 is a lot. It would work, but I argue that there is a simpler statistical test that you can just use out of the box without worrying about permutation tests. So if I want to see whether some Go annotation, say DNA binding, is associated with a list of genes, say 1,000, which are marked as ALS risk, What test can I use?
Maybe you need to, for example, specific Go annotation, you check this enrichment in the entire, I don't know, list of genes,
Uh-huh.
of, I don't know, women, and then check how many times it, like, what is the average enrichment of this Go annotation and what is average enrichment in your specific.
So you say comparing the average enrichment between my group and another group?
Or, no, the entire.
How would you measure enrichment? Like what kind of measure?
This is how you measure enrichment, right?
How?
Like, I will measure it like if in my I have 1,000 genes and I have
Okay.
specific X Go annotation 1 time, like 100 time, okay? So it's 10%, right?
Okay.
Then in the entire genome, let's say we have 20,000 genes and it's coming, I don't know, 1,000 times, so it's a different percentage. And then I don't know. This is enrichment, right? How do you define enrichment?
That's what I'm asking you. How do I find enrichment?
So I would like the percentage of time or the fraction of time it's appearing in this group versus the entire.
So it says just a proportion. I just want to make sure I'm clear about what you say. So let's say we have 1,000 genes which are associated with ALS, of which, say, 100 also have some Go annotation.
Mm-hmm.
So
how many times Go annotation? If
let's say that this
if
Go.
it's 10% of it's appear 10%, this Go annotation appear in the 10% of your list of genes. But it's also appeared in the 10% in the whole genome, so it's not enrichment. It's
Right.
just
Okay,
the
so you
number
want
of
we want.
times. So you need to compare like the fraction of it in your
Okay, I got it.
list to the like.
Okay, yeah, it's a reasonable measure. Any other ideas?
I think Fisher exact test. Like if
Right.
If we look on specific enrichment, so you can have the list of enrichment and not enrichment,
The category.
and the category is associated with ALS or not associated with ALS.
Okay, so yeah, you're right. We can use Fisher's exact. Can everybody see that? How it works? So we can simply define the genes by 2 categories. One category would be either ALS or not ALS. So we would have 1,000 which are ALS risk and 19,000 which are non-ALS. And then the second category would be whether they have or don't have this Go annotation. And then Fisher's exact would simply give the result. So that would work for a single Go annotation. Now, if we wanted to go over the entire Go annotations and get the list, what should we do then? Sorry? Chi-square, you say? Okay, so you say we could do a chi-squared by, so how do you structure the table?
So basically, as you said, but like for each Go term, just multiply it. So
It's multiply. So you want one row per Go annotation.
yeah.
Okay, I see a problem with that. Can you understand? Do you
Oh,
have an idea why it wouldn't work?
Overlap.
Overlap, right? So when we use chi-square, we need the categories to be mutually exclusive. Now, Go annotations are not mutually exclusive, so.
do a permutation on the theta.
Right. So what other strategy did we see in those cases? I mean, Fisher's exact works. It just works for one category,
So
for
you
one
do
value.
tons of them and then do some correction for multiple testing.
Yeah, okay. This is also a good strategy, right? We could do many Fisher exact tests, and then for each Go annotation independently, and then we could just correct for multiple hypothesis testing and get a Q value for each Go annotation. And then we actually know which Go annotations are enriched in this test. Okay, I hope this illustrates that it's very non-trivial to come up with the right kind of tests, because it's really confusing. We have like the ALS here, which seems to be relevant, but it's not really, just a bunch of genes. And we need to remember that we actually need to carry out the analysis on genes and not on people, even though we speak about a disease that inflicts humans. So all of these like considerations are super important, and it's very, very easy to get it wrong. So that's why I think it's important to go through many examples and really get to practice this kind of thinking. What about the next one? So we want to test whether Python programmers are more likely to publish highly cited papers. Now, I think it's self-evident, but suppose we suppose we came across some skeptic person and we wanted to convince them that it's actually true. How would we do that? Okay, so you say Fisher is exact with which categories?
So whether or not you're a Python programmer and what I mean, yeah, and how.
Or ANOVA.
Sorry?
Either ANOVA or t-test, depends on how many groups. If you have Python programmers and non-Python programmers, then you have the number of cited paired programmers. So you can do like an average or look at the median.
Yeah, we could simply do a t-test by comparing the number of citations of Python programmers and non-Python programmers. Or we could, by the way, also model Python programming as something continuous. So maybe not all Python programmers are the same. So we could measure like how many lines of code they have written and stuff like that.
The universe of that.
Yeah. Okay, what about the last one? We want to test whether a daily meditation routine helps preventing psychiatric disorders. What kind of tests?
What is the Fishers?
Fishers, which categories?
Daily meditation or not, and psychiatric disorders.
Okay. That's an option. Any other way?
You can
Do you have any
measure the
degree?
number of hours of meditation.
Right, yeah. We could also model it.
Measurements.
Yeah, exactly. We could also
You can
model
also,
it
like,
as something
measure
continuous.
Psychiatric disorders as well, like tests in which you have scores, psychiatric scores for each test, and then you can.
Exactly, yes. So most things can also be modeled as something numeric. Okay, now suppose I got a significant result. Would it prove what I wanted to prove? Right, notice the kind of language we use here, which is different than the kind of...
Hypothesis didn't prove your reputation.
Exactly. Notice that the word helps implies some causal relationship, okay? So in the other cases, we simply spoke about association, which was fine. But here, if we actually want to test whether meditation really helps to prevent psychiatric disorders, it wouldn't be sufficient just to look for associations. Ideally, we would want to make some kind of experiment, maybe an RCT or something. Okay, any questions? Okay, let's look at more elaborate examples. So here we have a researcher which wanted to find out which genes are overexpressed in cancerous lymphocytes. So she RNA-seqed 25 samples from a healthy patient and 25 samples from a diseased patient. What statistical test should she use?
You can do t-test and then correct multiple comparison. You do ANOVA and then post-hoc. So ANOVA.
Okay, so you say do a t-test of each of the genes and see whether you have more expression in those 25 samples than in those, and then correct for multiple testing. And then you would find the overexpressed genes in cancerous lymphocytes. Would it be a good conclusion?
The altered gene in cancer.
Sorry?
The altered, either over or underexpressed.
Right.
You can do directional t-test then.
Exactly, but the question is, would it prove that those genes are actually overexpressed in cancerous lymphocytes?
No, not that much.
What would it prove?
You prove they are different. It doesn't prove that
Different between what?
they're different than the healthy one. You prove that the sick people have I don't know, some sort of different RNA expression in their tissues, but it won't necessarily say that the cancer is the cause. It could be that the cancer caused it, or that's what caused the cancer.
How many patients do we have here in this experiment?
50.
No, read it carefully.
Oh, 25.
Two
One. Oh,
patients.
one.
Two patients.
We have 2 patients, okay? I mean, So one of them is named John, and the other one is named Michael. And if we see that John's genes are more overexpressed than Michael's, clearly it doesn't prove anything about cancer. Okay, it could just be these two specific people. So again, I tried to lay out these traps for you. So you notice that it's actually important to have the samples independent. Okay, these samples are not independent, and so you cannot really conclude anything to the population that you care in general. Now you can make some conclusions about John and Michael, but arguably it's not the thing that this researcher wanted to do.
But if you do t-test for dependent measurements, no, it's not the same because it's not
Okay, so notice, these are, again, very subtle things, but important to notice. So this is a really bad study design, and I think there is really nothing meaningful you can do with this data. You just need to collect the better data if you want to arrive at any meaningful conclusion.
What
Sorry, question?
would you be able to do? Like take lymphocytes that are not cancerous? Is there a
25
possibility to find
No,
more cancerous.
if you actually want to arrive at conclusions about cancerous versus non-cancerous patients, you would actually want to collect independent samples from different patients. So you can take 25.
different patients? This is the problem.
Sorry?
These are different patients and this is the problem.
No, these are not different. You have 25 samples from the same healthy patient, and 25 samples from the same. You need more patients, yeah. And if you have multiple samples from the same patient, you cannot really use them within a t-test or something like that, because those tests assume independence. And if you have them from the same patient, they are not independent. Okay, let's see more stories and really try to think critically and like what's going on wrong in those stories. Okay, so what do you think about the following one? So we have a pharmaceutical company which is studying the effect of a new drug in mitigating symptoms of Alzheimer's disease. Now, after testing a large number of patients in an RCT, a randomized controlled trial, t-test found a very significant association, so you see it here, very significant, between administration of the drug and success in cognitive tasks. So impressed by the effectiveness of the drug, the company's board of directors decided to further fund the research projects. What do you think of their decision?
Depends on the effect size.
Okay, so what do you think? It depends?
It could be a very significant statistical phenomenon, but if the difference in their cognitive functions was small and the effect size, then it's not worthwhile to
Exactly.
pursue
So
as a drug.
exactly. So again, the board of directors have confused significance with effect size. So the fact that you got a very significant p-value doesn't mean that the drug is so effective. It could be very, very small effect size. It could just be that they have a very large sample size. So there is this concept of like too big to fail when we deal with very large samples. So if we take 1 million people, like even the smallest kind of effect size would make up a very significant result. Okay, so again, it's very important not to mistake them. Okay, what do you think about this one? So a lab studies the effects of sugar-rich versus fat-rich diets on mice. They designed a study where 10 mice were randomly divided into 2 groups of sugar and fat-rich diets. During 5 days of experiment, several physiological measurements were taken from the mice, ending up with a total of 50 samples. After examining all taken measurements, they found, using t-test, that fat-rich diet is significantly associated, and you can see the p-value here, with elevated blood pressure. For multiple hypothesis testing?
Yeah.
So what kind of multiple hypotheses you have here?
We can do what?
So
Sorry?
you say they need to correct for multiple hypothesis testing. So what multiple hypotheses do you have here
They
that you need to correct?
I don't know how many measurements did they take. Several.
Okay, yeah, it's right. So it says that they took many physical measurements, but now they only report about the blood pressure. But maybe it's one of like I don't know, 100 different measurements, and then it's not so significant. So you're right, they need to account for multiple testing correction. Anything else that might be wrong here?
It could just be constant.
Sorry?
Maybe it's significant, but because you give it's both fat rich and sugar rich, it's like it's expected to happen. It's like expected to happen. It's not the significance is not like you need to compare a normal diet to
Okay.
blood pressure.
Okay, so you say that they don't compare to something normal. So we don't actually have a control group, we just have like 2 case groups, and then the conclusion that fat-rich diet is significantly associated with blood pressure could be wrong. I mean, it's different than sugar-rich diet, but maybe fat-rich diet is like normal, and sugar-rich diet is the one that is unusual. It could be. So yeah, that's another problem. Anything else?
They don't report the effect size.
Right. Okay. They also don't check the effect size, but here, I mean, yeah. Okay. They only report that it's significant, but you're right. It's better to also report the effect size. What else?
Maybe the samples are dependent because from 50 samples, it means they took several samples from the same mouse.
Exactly. That's the exact problem we spoke about before. So again, we only have 10 mice which were measured during 5 days. And then we apply a t-test, which, to remind you, a t-test assumes that the measurements are independent. But that's not the case. We have, for each mouse, we have 5 different measurements of the same mouse. It's not independent, and we cannot really use a t-test. Okay, is it clear?
So you can use just from one.
It's very problematic. So definitely not a regular t-test. There are some versions of t-test that can work for dependent samples. We didn't speak about it, but you do have.
Like dependent t-test for this one.
Also not exactly, because it's more complex. Like you have 5 different measurements. I don't know, maybe. I'm not familiar with the version of t-test that works here, but maybe there is.
Usually you do want something like this. So for all the samples from one day, I'll compare between them, then I'll compare between the samples of the same mouse, then I'll know if like you want this type of dependent
Yeah.
samples sometimes.
Sometimes you want it, yeah. It depends how you use it. So yeah, sometimes it can help you to have,
You could use this t-test on one day sample,
yeah,
then it will be dependent.
exactly. Or we could take some aggregate. Maybe we could take the mean or the median per 5 days, and then we would have like more stable kind of measurements, which we could then compare between the 10 mice. So for 10, it's probably not sufficient to have statistical power, but if we had more mice, you know, it could be useful to have more than one measurement. But it would be wrong just to put it within a t-test as if it were independent. Okay, a third one. So here we have a student which wanted to find out whether proteins with PDZ domains are likely to have other domains as well. So she built a contingency table with 2 columns, presence or absence of PDZ, and hundreds of rows for other domains, each cell counting the number of proteins with that combination. She iterated through the rows and ran Fisher's exact test for each of the domains. She adjusted for p-values for multiple testing correction using FDR and found several significant domains. What do you think?
Maybe it's not that dependent. Maybe the PDZ domain itself is associated with other domains usually. So, but that's what you start to find out from.
Yeah.
It sounds smart. Sounds
So-
like she did everything.
It sounds sound to you. Yeah, actually it is a sound. So in this example, it's actually fine. I wanted to test you. Just one thing to notice here is that we actually counted correctly. So if we build 2 by 2 kind of contingency tables with presence or absence of PDZ and presence or absence of other domains, then it's fine. Just make sure that when you do it, you don't start with a bigger contingency table that has a row like for each domain and then sum it up, because then the arithmetics wouldn't work because it's not mutually exclusive. So like for each combination, you need to do the calculation from scratch if you want it to be properly done. In this case, it doesn't make sense to build a big contingency table. So, I mean, because of the overlap, right? OK, next one. So here we have a student that wanted to determine whether a list of 182 genes predicted as targets of a specific microRNA are associated with specific tissues. So he built a contingency table for all human genes with 2 columns, targets versus non-targets, and a row for each tissue. He ran a chi-square test after validating the expected frequencies were appropriate and found an association.
He didn't do the correction from multiple tests.
Multiple testing correction. Why would you need multiple testing correction here?
Because he did many different experiments, like many tests.
No, he just ran a single chi-square test. So he built this very big, for all the tissues together. So he built a huge contingency table for all the tissues and then ran a chi-square test. Now you're right, you cannot use this to tell which are the tissues that are different, but now it does have a p-value saying that there is a significant association
Why did you do it
somewhere.
for the entire genome?
Sorry?
Why did you do it for the entire genome? If he's interested in 180.
Yeah, but only like, you wanted to test whether those 182 are associated with tissues. If we only looked at them, you would just have numbers, but you wouldn't know what to expect, right?
Expected value.
Any idea what might be wrong here? I will give you a hint and say that we already discussed it a few times on like the last 15 minutes. So
Genes are not expressed independently.
what do you mean?
So usually genes affect other genes or transcription factors or whatnot. So if one is being expressed in a tissue, the other should be expressed as well. Maybe they're not independently one another.
So actually that the genes are dependent on the tissue dependent on the genes that are expressed in it.
But the tissue is like the category, and what you look at is genes. So you want to see whether you have some enrichment of the genes in some combinations. Now, I'm not even sure what it would mean for genes to be dependent or independent. If there are different genes, then you look at all the genes. So you have like the complete sample.
It's like binary, is it being expressed or not? It's not how much it's being expressed.
I mean, you could model it to something continuous. But here we just decided to go simplistic and just, yeah. For each tissue, is it expressed or not? Okay, I'll give you a hint. What's wrong with defining the tissues as a category? What kind of property the tissue doesn't hold that you would need it to hold to use a chi-square test? Are tissues mutually exclusive with respect to genes? Okay, so we look at genes that are associated with tissues. Now, the same gene could be expressed in many different tissues. So it's important that the kind of thing that you're actually counting within your contingency table, so if it's patients, then patients. If it's genes, then genes. Whatever you're counting, it has to be mutually exclusive between categories. So when it comes to genes,
Like all the categories in the columns have to be mutually independent, but not mutually independent with the rows.
right, so between the rows and columns, you don't need anything. Like that's the thing you're testing. You're testing if there is some association. But between the rows and between the columns, you need it to be mutually exclusive, okay? So if we count genes, we cannot afford to have the same gene counted multiple times within our table, okay? We need to decide which row it belongs to. Now we could look at only at genes that are expressed only in one tissue, and then it would be fine. Then the question would be somewhat different. We would ask the question only about genes that are exclusive to one tissue, and for them we might say, okay, now we see some tissues for those kinds of genes that are different between targets and non-targets of these microRNA. But in order for it to work in the first place, we must have this mutually exclusive thing. Now, it's a bit confusing because it really depends on what is the thing that you are counting. So you can think in other stories in which using tissues is perfectly fine because, I don't know, maybe if you count bruises of patients, so when you see which organ they have a bruise on, then it's mutually exclusive. You cannot have something on two different organs. But if you're talking about genes and which tissues they're expressed in, then it's not necessarily mutually exclusive.
But if you're just doing one tissue versus the entire, the entire.
One versus rest?
Yeah.
Yeah,
You
then
also
then
have
it
that
would work.
rule?
Yes.
It's also not mutually exclusive.
Mm.
You're counting the same gene here and there.
No, but you can define it as so. You can say, okay, either you take genes that either are expressed in this tissue or not expressed in this tissue. So then it's really mutually exclusive. And you can do it using multiple Fisher exact test and then multiple correction. It would be fine. But again, you need to define it properly. You need to make sure that what you count is actually mutually exclusive.
So you could use that method to do the same experiment for each tissue versus all the rest.
Yeah. Yeah, then it would be better to use Fisher's exact because you have a 2x2 case. Okay, what about this very long example? So we have a researcher which wanted to know if protein phosphorylation is associated with cellular localization of proteins. So she built a 2x2 contingency table with rows corresponding to phosphorylation status, either phosphorylated or not, and columns corresponding to protein localization, nuclear protein or not. She iterated through all the reviewed UniProt records of eukaryotes, and for each record, iterated over its sequence. For each amino acid, she increased the value of the corresponding cell in her table. So if this amino acid was phosphorylated or not, and if it belonged to a protein that was a nuclear protein or not. And at the very end, she ran Fisher's exact test and after obtaining a significant result, concluded that nuclear proteins have a tendency to undergo phosphorylation. Yeah.
Not all the amino acids can be phosphorylated, so she needed to take only the fraction of the amino acid that.
Okay, that's a big problem. Okay, so it's kind of silly.
Option to be phosphorylated.
Right. It's kind of silly to look, for example, at the arginine residues, which just cannot undergo phosphorylation. So it would clearly bias the results. Anything else?
She's counting more than once the same protein.
Sorry?
She counting the same protein more than once. She counts the phosphorylation sites, not the protein.
Yeah, so you could seriously argue whether the thing you're counting here is really independent. So it will be really dominated by proteins that might have more than one phosphorylation site. It's true, yeah. What else?
themselves are duplicated? Perhaps?
Yeah, that's also a big problem. Okay, so she looked at the entire
Eukaryotes.
proteome of eukaryotes. So, and we know that we have lots of homologs between different species. So if you look at some protein like, I don't know, hemoglobin, you could have it like virtually in all of the animals, and you would count it many times. Again, breaking this assumption of independence in some ways. What else?
Some protein can be in the nucleus and the cytoplasm.
Yeah, that's right. But again, you could define it as somewhat mutually exclusive. You could either say a nuclear protein or not a nuclear protein.
It's possible that some of the proteins get phosphorylated because of the rotation and not get phosphorylated.
Right. So you're right. We cannot really conclude anything causal about it, so we don't know what causes which. But notice that she didn't really make any kind of causal conclusion here. She just said that it seems to be the case that they have a tendency to undergo phosphorylation. But what do you think about this conclusion? Like, suppose that we have dealt with all of the problems that you raised. So we limited the kind of analysis just to relevant residues, and we got rid of all the redundancy. Would it then be okay to make this conclusion about proteins?
What is exactly her null hypothesis? That it won't be more phosphorylated?
So the null hypothesis. So the null hypothesis with Fisher's exact test is that you have independence of the rows and columns. So in this case, that there is no association between being nuclear protein or not and undergoing phosphorylation or not.
I think she didn't really.
Yeah.
I think I would not say have a tendency to undergo phosphorylation, and more that somehow phosphorylation happens more. It's like
Yeah.
appears more often.
So actually, phosphorylation could occur and then desphosphorilate. So maybe you see a dephosphorylated protein, but it did go phosphorylation when it was, I don't know, in a different in a harsher conditions, or now it's a harsh condition. And
Yeah, it's also very context dependent. And it could be like phosphorylation is something that happens for some proteins some of the time and not all of the time, and it depends on like you define it. But you could go with the more simple kind of definition and say that if at some point in some conditions it undergoes phosphorylation, then for the sake of this analysis, we consider it to be a phosphorylated protein.
but you don't see it right now. Like maybe
Yeah.
But with this wording, you somehow.
Yeah, so you're right that this conclusion, again, might be a little bit off because it speaks about phosphorylation in general, and it could be that we deal with a protein that in some context undergoes phosphorylation, but that's not right now.
With
Yeah,
this wording, you define a causal relationship, no?
sorry?
Like a causal
Causal, yeah.
relationship with this wording have a tendency to undergo.
Yeah, I don't think, I mean, in the end, I guess it's a matter of like how you understand the language. But I think that the wording tendency to undergo generally doesn't imply causality. So you could say that tall people have a tendency to also have red hair, without implying that it means that being tall actually causes their hair color to change. You just notice this kind of phenomenon. Yeah.
So she concluded direction, and her test is unidirectional. So
But again, she used the wording tendency to undergo, which it's quite symmetric actually. So saying that nuclear proteins tend to undergo phosphorylation is the same as saying that proteins that undergo phosphorylation tend to be in the nucleus. It's the same.
yeah, but then compared to what? Cytoplasm and
Compared to proteins which are not in the nucleus, or compared to proteins which are not phosphorylated.
But shouldn't it be phosphorylated or not
Yeah, I
in
don't
the
understand
nucleus?
amino acid. Oh, I'm not asking the
sorry?
same thing.
Shouldn't it be phosphorylated or not in the nucleus, not versus other areas of the cell?
I'm not sure I follow.
It's like if she tested phosphorylated or not, nucleus or not.
Right.
Shouldn't it be more exact to say phosphorylated or not phosphorylated in the nucleus to see what's the portion of the phosphorylated in the nucleus itself, not
Right. Yeah.
in
Again,
other part of the cell?
I mean that really depends of like what kind of biological conclusion you want to arrive at. So it's right that if you actually want to see what's actually going on within the nucleus, you would have to take some snapshot of the cells that are actually within the nucleus. But here we have like more mild goals. We just want to see whether in general we see a tendency of proteins which at some phase are in the nucleus and proteins which are at some phase undergo phosphorylation, and whether we see some correlation between those groups.
I don't think Fisher is the right thing to do here.
Okay, yeah, more ideas?
I don't know. Maybe I don't understand exactly what she did. If she's checking if a protein is phosphorylated or not, why is she checking each amino acid? It's enough that one amino acid is phosphorylated.
Yeah,
I don't understand the relationship here between the number of phosphorylated.
Okay, yeah, you're right. So here you got it. So again, it's very subtle, but you notice that the kind of samples that we count within this table, we don't count proteins, we count amino acids. So amino acids are the entities on which we perform this analysis. But then the researcher went and took this kind of result and concluded something about proteins, which are a whole different entity. So when you do analysis about amino acids, you cannot really conclude about proteins. And when you do analysis about proteins, you cannot conclude about amino acids. These are different things. And it's very confusing because like on an intuitive level, it seems like very much related. So we tend to identify the proteins with their amino acid sequence, forgetting that they are actually two different entities. And it's actually important that when you do statistical analysis, you don't only look that. We spoke earlier about selection bias, about how when you perform like kind of analysis on one group, you cannot really conclude things about different groups. Now, when we speak about different groups, we need to make sure that we are dealing with the same variables and the same distribution, and also very crucially, the same entities. So if you are making an analysis about amino acids, it won't tell you about genes or proteins or other kinds of entities. And you need to be very sensitive to these kinds of definitions, which can be very confusing. And yeah, the general theme, I hope these help to really convince you that it's not trivial to be skeptic enough in the face of such very easy mistakes that can pop up into your analysis. Okay, so I think here we really are going to conclude this discussion. I will just say that even though we covered really a lot, that's really far from really covering the whole space of like this entire topic. And there is really lots of tests and theories, some of which we mentioned, which we didn't really discuss. And of course, if you're uncertain about something, like I said, it's very important to stop for a moment and try to think it through. And if you're not sure, it's always a good idea to ask your friend or your supervisor or some expert in statistics. And there are many relevant subjects which we didn't really discuss in those 2 weeks, which still I think are very relevant. The first one is multivariate analysis. So the case all the tests we mentioned were just in the case where we have 2 variables. So essentially, we spoke about many kinds of association tests between 2 variables, but in many cases, we have more than 2 variables. And it's very common that we want to account for more variables. So it's very common if we are worried about some confounding, then we want to take those potential confounders into account and see if our result still holds given those confounders. And for that, we need to use more advanced tools, which are called multivariate analysis. Now, we didn't speak about it today, but we are actually going to cover that topic next week. So that should be OK, at least some kind of analysis. A second important topic that we didn't really touch on, we just mentioned, is causal inference. So we did say that correlation does not imply causation, but we didn't say what exactly does imply causation. Again, this is a very this is a huge topic, actually a very modern topic. So it's something people started to systematically address only like on the last decades. And there are some courses at the university or other places that you can take if you're interested more about that. There is one book which I really recommend. I think it really is one of the best books I read. It's called The Book of Why by Judy Aparo. It's a really nice book, I think, especially if like you want to think about it in a more systematic way. It really helps to think clearly about those issues, and I think it's also a very accessible book. So it's really it's very nicely written, because on the one hand, it's not like a popular science book. It is made for like for people with more expertise on the topic, so people that actually work with data. But on the other hand, it's not like a very complicated textbook that you like need to you need to spend hours trying to understand what exactly it means. It's something that you can easily read before you go to sleep, and it's not something too intense. And I think it's really a nice read, which really sheds light about this topic. Another topic which we, again, didn't really discuss was things related to information theory. So it did pop up a little bit when we spoke about logos and entropy and stuff like that, but we didn't touch on it too much. Again, that's a whole different field from statistics, and just it happened historically that this is different kind of people which work on this, and they have like their own different kind of definitions and way to look at things, which I think in many cases can be interesting and like be very complementary to the kind of statistical way of looking at things. So we did speak a little bit about entropy, but also things like mutual information and KL divergence and stuff like that. Again, this can be an interesting other way to look at data, and I think this can be relevant if you want to look into it. And then there is also the topic of machine learning, which again, it's very similar to statistics in many ways, but again, historically it was developed independently and used for different kind of use cases. And that's a very powerful tool that many scientists in many disciplines start to use. So it can be either, for example, predictive models. So you can think about regression, so linear regression that we spoke about, and logistic regression that we are going to speak about next week. You can think about it as some kind of simple predictive models. But then you can have even more kind of sophisticated, so decision trees, random forest, stuff like that. Or it could be clustering algorithms, which are, again, very useful in many domains. So again, we won't really speak about it, but there are many courses and many online resources that you can check out. And the good news is that since you're already kind of experts in Python, it shouldn't be too hard for you, because Python is really the dominant language when it comes to machine learning. So you have very strong packages like scikit-learn and others. And yeah, since we started this discussion with a quote about the 3 kinds of lies and statistics being the worst, I thought it would be appropriate to finish with a quote. And yeah, I think we'll stop here.